So now we are going to begin with lens, the very important topic of ophthalmology, the lens. Now, basically we will start with the anatomy and the metabolism, okay, part. So anatomy basically first. Now, the introduction to the lens. What is the color of the lens, blood supply, nerve supply, and the shape? We all know, just a quick revision. Like the color is transparent, okay. The blood supply, the lens is avascular. The nerve supply, no innervation to the lens, okay. Only the muscles are responsible for the movement. The shape is biconvex. Remember this, okay. So the important thing over here is the shape. So this is your lens, okay. We have drawn a line passing to the center of the lens, okay. This divides it into anterior surface and the posterior surface. So these are the two surfaces of the lens. Now a brief about the surface. So the anterior surface of the lens is lesser convex. Remember, overall the lens is convex, but the anterior surface is lesser convex and it is flattened, okay. Now the radius of the curvature in the anterior part is 10 mm. The posterior surface is more convex, okay. Remember, the anterior surface is less convex and the posterior surface is more convex, okay. Now the posterior surface is curved due to this concavity, okay. What is the radius of this curvature? It is 6 mm, okay. So anterior surface radius 10 mm, posterior surface radius 6 mm. Now the equator diameter is 10 mm, okay. So you can see over here the equator, its diameter is 10 mm. Now another thing, anterior posterior diameter, the anterior posterior diameter of the lens, what is it? So the posterior or the site of the lens, which is also known as patellar fossa, okay. Now the refractive power of the lens is plus 16D to plus 19D, okay. Now the refractive index of the lens at the center part is 406, okay, 1.406 approximately, and the part of the lens with maximum refractive index is the center part, okay. Now the peripheral part, what is the refractive index? It is 1.386. So basically in the anatomy we missed a point, that was the lens is developed from the surface ectoderm. So the lens is derived from the surface ectoderm. Remember this, students, embryological point of view. Now the lens and the associated structures. So what basically associated structures you can see at the lens? These are vitreous base, you can see. Now Vagart's ligament, Burger's space, Cloquet canal, and the space of Martigiani, okay. These are the associated structures that you see posterior to the or near to the lens, okay. These all structures, it has been marked by the red color, okay. Now we'll look them individually, all of them, okay. Now beginning up with the Vagart's ligament, okay. So the Vagart's ligament, it is basically the attachment between the posterior surface of the lens and the anterior surface of the vitreous, okay. So this is the vitreous, okay. So the anterior surface of the vitreous and the posterior surface of the lens, these are the two attachments which forms Vagart's ligament, okay. Now the next comes is Burger's space, okay. What is Burger's space? It is also called as retrolental space, okay. Burger's space also called as retrolental space. And what is this space? this is a space between the lens and the vitreous. Okay, so attachment between the lens and the vitreous, it is Vagat's ligament. And so now Burger's space, what is Burger's space? As we studied, it is the space between the lens and the vitreous tuber. Okay, the next and the important one comes the Clockwork's canal. Okay, so Clockwork's canal, what is Clockwork's canal? This canal which you can see over here, this is Clockwork's canal. Let us study in the theory. Now basically there is no arterial supply for the lens, but at the time of embryogenesis there is a arterial supply to the lens, that is called as, that is done by the hyaloid artery. Okay, so the hyaloid artery supplies the lens during embryogenesis. Okay, after that it gets diminished. Now the Clockwork canal, or you can say it the hyaloid artery, this get diminished after 8 months of gastration. Okay, so after 8 months of gastration the hyaloid artery gets diminished. Okay, if there is the persistence of this hyaloid artery, then this leads to Mittendorf's dots. Okay, what is Mittendorf dots now? So it is the remnant of hyaloid artery on the posterior surface of the lens. Okay, important point, posterior surface of the lens there is remnant of hyaloid artery. Okay, the next comes is Bergmann's papillae. Now what is Bergmann's papillae? It is the remnant at the optic disc. Okay, so this is at the posterior surface and this is at the optic disc. That's it. Now you can see several layers of the lens over here. Okay, these diagrams. Now what are the layers of the lens? So there are basically 4 things. These are outer layer, the anterior epithelium, there is the cortex, and there is a nucleus. Okay, which is divided into 4 zones. Let us study the outermost layer, also called as the capsule of the lens. Okay, now the capsule is the thickest basement membrane at the body part of the lens, and it is made up of type 4 collagen. Okay, very important, type 4 collagen. Now thinnest portion is at the posterior pole. So basically capsule is the thickest basement membrane in the body part. Okay, and the posterior pole is the thinnest. The thickest is the posterior pre-equatorial zone. Okay, so posterior pre-equatorial zone, this you can see by the mark black line. Okay, and what else? Capsule, so anterior capsule you can see over here. Okay, now the anterior epithelium. Okay, so let us find an anterior epithelium in the diagram. Here is the anterior epithelium, which is marked by pink color. Okay, you can see what is this anterior epithelium. So the anterior epithelium have 2 types of cells. These are A cell and the E cell. Okay, the A cell is responsible for the metabolism and the E cell are for the new lens fibers, as they are capable of mitosis. Remember this. Okay, very important point. The E cell are responsible for new lens fiber formation, as it is capable of mitosis. Okay, now the posterior epithelium, it is absent as during the embryogenesis. Okay, now the posterior epithelial cell elongates to form the primary lens fiber. Okay, so during embryogenesis this posterior epithelium is absent, primary lens fibers are formed. Okay, after that, now coming up to the cortex part. You can clearly see this whitish part in between. It is nothing but the cortex. Okay, let us study about the cortex. The cortex is the young lens fiber, and there is outer cortex. Okay, so young lens fiber also is its a outer cortex. Now, the nucleus of the lens. The nucleus of the lens are composed of four bodies. These are adult, infants, fetal, and the embryo. Okay, so in such stages, the nucleus is being formed in a different manners. Let us study the four zones. These are E for embryonic zone. Okay, so primary the lens fibers are present in the embryonic center, and it is the oldest lens fiber. Okay, because it is formed at very first. Now comes the fetal zone. Okay, then infant and the adult. So these are the secondary lens fiber. So what is the mnemonic? EFIA. Okay, EFIA in this sequence. Okay, so embryonic, the first one, then comes your fetal, then infant, and the adult zone. So in such a manner the lens is growing. Okay, so the nucleus of the lens is growing. Okay, now there are some sutures in the lens. We have studied this. Now these are formed at the fetal period. Okay, so the sutures of the lens are formed at the fetal period. The anterior sutures are Y-shaped and the posterior sutures are inverted Y-shapes. Okay, so just a slight difference. These are Y-shaped and these are inverted Y-shaped structures. Okay, so that was all about the topic. In the next session we are going to discuss about the proteins in the lens. Okay, very important topic in the part of lens.